So good morning to those of you who survived morning tea and come back. Sorry, got to allow for my height here. Firstly, I'd like to thank Raji very much for the invitation to come back to Hyderabad. Had a very pleasant week here and I've been enjoying the meeting very much. I've agonized a little bit about this talk and what I should try and cover. And really listening to many of the presentations that we've heard yesterday and this morning, I decided I'd shuffle things around a little bit and do this somewhat differently from I originally intended. So I hope this is going to work. I need to sort of start by setting the scene a little bit about the species and the problems that I want to talk about. So I'm going to talk essentially about wheat. I'm going to talk a little bit about barley because barley serves as a nice model for wheat as a diploid, and there's a lot we can learn from barley in studying and analysing wheat. I think the important thing to remember is that globally the yields of wheat are generally quite low. This table up the top left there shows the four major cereals, and you can see that the average global yields of wheat are about 3 tonnes per hectare. If we remove the irrigated wheats from that, then the average global yields are well below 1.5 tonnes per hectare. We also know that the record yields for wheat, which come from New Zealand, were slightly over 15 tonnes per hectare. So by and large, in most of the environments where wheat is being grown, somewhere between 80 and 90% of the yield is being lost due to a whole string of factors, and probably most important amongst those are basically moisture stress or water availability. Now, moisture stress is, of course, an extremely complicated stress that has a very large number of components. And this cartoon up here is really to try and illustrate the sort of factors that are going to be limiting production in these low-yielding environments where wheat is grown. We not only have the direct issue of water availability, in other words, drought stress, but drought stress usually comes combined with heat. And the combination of drought and heat, as I'll show you later, is a very different type of stress from drought alone. In addition, in many of these environments, the plants are exposed to hot, dry winds, which, of course, increase the evaporative demand dramatically on the plants. If we go down below the surface of the soil, there are a whole lot of other issues that are going to impact upon productivity. In particular, there are lots of pests and pathogens that will be damaging and grazing on the roots, again, reducing the ability of the plants to access moisture. And in many of these environments, the soil moisture or at depth, there are a whole lot of other problems. I live and work in Australia, and southern Australia in particular, where the groundwater is essentially saline. So there's really not much benefit to the plants to try and tap moisture at depth. It may not only be salinity that's a problem, but you'll hear from Tim Sutton later on, there are problems of boron toxicity, nutrient deficiencies, and a whole range of other things. And we also know that the soil structure can have a big impact upon the ability of the plants to extract moisture. So by and large, wheat is facing a really miserable life to put up with all of these stresses. As I said, 80 to 90% of the yield is lost in most production environments due to the problems of accessing moisture. Okay, so in this sort of scenario, is genomics of any particular use or help? And what I want to try in this talk is go through some of the issues and problems that we face in trying to, I think, apply genomics technologies to address and tackle some of these straits. We know that genomics, and we've seen very good examples already about its relevance in developing resources and analysing germplasm and material, in supporting gene isolation and understanding biochemical, metabolic, and other processes and pathways, as a tool for trying to explore and utilise allelic diversity. And then we also know that there are a number of quite sophisticated methods available for deploying genomics information, either through conventional breeding or through genetic engineering. Probably central, though, to many of these features is the way in which we go about integrating the various technologies and systems. And again, that's a point that's been repeatedly raised during this meeting so far, and I think is a point that we're going to hear a lot more of. I suppose in the traditional view of where genomics sits and how genomics operates, it's essentially a combination of a very large number of platforms and technologies. And this diagram here is really meant to try and summarize that. And I think it's sort of a pattern or a process that many of you will be aware of. We understand that the process really starts with the identification of a trait or a characteristic of importance, a knowledge of the germplasm and genetic variation that might be available. That variation can be used for generating segregating populations to try and analyse the genetics. We can go into a direct omics approach to try and tease apart some of the processes and pathways that might be operating. We mustn't forget the physiology that sits within all of this, and certainly when we start talking about stress tolerance, a knowledge of the physiological processes becomes very important. Then by and large we try and integrate all of this information together, again as you've heard, to develop models about how the pathways and processes are interacting and might be combining. We need to link all of this to the central component here, which is the phenotype. So do we actually know what we're looking at and measuring, and how do we characterize that? And that will be a recurring theme in this talk, and has been quite a theme in the presentations we've heard so far. The genotyping we know is now becoming reasonably straightforward, and we've heard about some of the advances in those platforms. We then try and combine all this together, use information across between related species to get at the genes, the pathways and the processes, and hopefully with that we have something that we can then transfer to the breeders, and with a bit of luck something useful will come out of all of that. So that's sort of the broad concept. But how does this really look in practice when we try and do the analysis? So I said I really wanted to talk a little bit about that phenotyping issue, and we've heard again this concept of a phenotyping bottleneck. I'd really raise the question about whether it is actually a real bottleneck, or that it's really an issue about how we utilise phenotyping information and analysis. When there are a number of options we have, particularly when, and you should remember I'm trying to focus on these highly stressed, these difficult environments, traits like drought, stress, salinity and so on. We have a number of options to try and deal with this and try and measure the phenotype. One is we can try and fix the environment. So we can use controlled environment facilities, we can use fancy automated greenhouses. We can monitor the environment, and we can try and include components of the environment in our analysis. We can use components of the phenotype and try and tease apart the different aspects of it. And then of course there are been significant advances in the way in which we go about analysing the data and information in both design of the trials, be they in a controlled environment or in the field, and the way in which we tease apart useful information, which in our case is the genetic information from those studies and analyses. So when we try and look at this though in a bit more detail, there are significant problems and limitations. And here illustrations of some of the sorts of phenotyping platforms that we use. The one up there where you're looking at single plants within pots in an automated glasshouse with imaging. We have a large bin system here where we can sort of try and get a bit closer to simulating the field environment. And then we have these sort of semi-controlled field sites where we're using rain-out shelters, simulating heat stress, whatever that might be. But it becomes quite difficult to work about the type of stress that we want to use and the way in which we want to deploy it. This shows sort of the problem that we face in, for example, if we're trying to use a pot base or a large bin system. Are we looking at a steady drought level? Are we looking at a moderate drought, a severe drought? How are we phasing the drought with the developmental stage of the plant? And how does that impact upon the way in which we go about assessing the phenotype and doing our genetic analysis? I thought one way of sort of showing the problems that we face in dealing with these platforms is to go through a couple of examples. The first example I'm going to show you is a very negative example, and it shows how some platforms are really not particularly useful. And here we were really trying to compare measurements we had on a series of genotypes in field and in the high throughput platform where we're doing imaging and so on. This is looking at drought stress to population. There are a couple of hundred lines that were looked at, and then we've got here just a whole series of traits that we can measure in the automated platform using a well-watered condition, a moderate drought and a more severe drought, and then this is a field situation. We have well-watered and droughted, and where you see red here, there's some sort of correlation between the traits. So on the surface it looks quite good. There are clearly a few traits that we're seeing in the platform that are also coming out with drought stress in the field. However, if we look at this, there's a fair spread in maturity, about a 20-day spread in flowering time between the lines that we used here. If we then remove flowering time as a variant in this, in other words, you look at a more narrow set of germplasm, suddenly we find our correlation between anything we see in the field and anything we see in that fancy, and I should say very expensive phenotyping platform evaporates completely. In other words, there is not a single trait that we could measure in either system that is correlated. So clearly, using that platform for assessing drought tolerance in wheat, and I need to emphasize in wheat because colleagues have got much better results in other systems, simply doesn't work, and it's not an appropriate platform to use. Having given you a negative example, let me give you a positive example as well, and looking at salinity tolerance. Salinity tolerance is a difficult trait to work in the field, in many respects even more difficult than drought, because you can't very well go and dump a truckload of salt in the middle of a paddock and then do your trials. You'll make yourself very unpopular with the local farmers if you try and do that. So you need to find sites where you have a spread of salinity. This is an example from work that a colleague, Stuart Roy, has been doing, looking at evaluating germplasm under a low salt and a high salt. This is just EM38 scans of the soil, and you can see, even though there's a degree of variability in those two sites, you can see this one clearly is a salt-prone site, and this is a relatively low salt site. And so we can screen large amounts of germplasm in these sites and get an idea of how they perform. So what happens then if we go and look at our fancy platform again? And here we've got two varieties that actually show only a relatively small difference in their salinity tolerance under the field trial. But Yitpi, this variety here, is clearly far more tolerant than Berkut. And in this platform we can measure growth rates of the plants at different levels of salt. We can then look at the differential. We can look at the way in which they're responding to salt over a period of time. And you can see here clearly Yitpi is showing relatively little effect on its growth and development under salt stress, whereas Berkut is suffering quite badly at the high levels of salt stress. And for this measure we get extremely good correlation with the field performance of lines. So for salinity stress, the platform works very well and has been used for quite detailed genetic analysis. Similarly, a number of other stresses, for example, nitrogen use efficiency, the platform also works very well. But as I said, drought and heat, we don't find a good correlation. Okay, so for these other stresses then, we are really very dependent then on using phenotyping in the field and analysis of field data. But clearly again, if the key trait we're trying to work with is yield under stress, obviously that's going to be a very complex integration of a whole series of pathways and processes that are occurring over time. And what we're really trying to tease apart is, I suppose, the genetic component of those different aspects. I've already showed you the significance of something like maturity, and I think probably all of you are aware of the impact of maturity on trying to analyze and measure something like drought tolerance and drought stress. Okay, so one of the ways we tried to tackle this problem was firstly with the selection of the germplasm that we worked with and the analysis. And so we've been now working for quite a few years, probably almost 15 years now, on a series of populations that were developed from lines that were all bred for the same environment. They were all varieties, commercial varieties that were developed and released in the southern Australian environment, but they differed in the way in which they responded to severe stress. So here we're really just looking at a whole range of field trials of 3 of the parents that we've been working with under a range of different stressed environments. And these are environments ranging from 2 tonnes down to 1/4 of a tonne per hectare. So these environments down here are really, really tough. In fact, by and large, in Australia at least, it's regarded if your yields drop below 1 tonne, then basically it's not been worth the trouble. But you can see now under severe stress these varieties respond differently. They're all yielding fairly similarly in what we regard as a good site. Under severe stress they're responding differently. Some are able to maintain their yield more effectively than others. So what we tried to do was to tease apart the genetics and the control of this pathway and these processes. And to cut a long story short, one can go through a fairly standard QTL mapping analysis. I should actually just say that for all of the parents that were involved in the study, and there were a total of 6 parents that we used, we basically threw everything that Omics has available. So the parents were analysed for metabolite profiles, a whole range of different tissues, a whole range of different stresses. We did proteome profiles, transcript profiles, you name it, we threw it at these parents to try and understand as much about the processes as possible. What we can then do is then start doing our genetic analysis and combining some of this data. This is an example of one of the regions we're interested in. This is on chromosome 7A. We had a whole range of performance traits from the field trials that mapped to this region here. And you can see here we also had a great string of metabolites that came from when we did the metabolite QTL mapping that also sit within that. So obviously a region of considerable interest to us. When we start teasing apart regions like this, though, there are a number of important points that that really come out of that analysis. So this is essentially the QTL scan across that region, and what we'll try to do is each of these plots represents the mapping in a different environment. And I've just tried to sort of show the three main markers in a 2 centimorgan region that sort of covers this area here, and the performance of the lines or the mapping in those different environments. The key thing here is that in some of these environments up here, we're looking at about a 14% yield benefit associated with the allele derived from RAC875. In these environments down here, we're looking at 2% or less. I should say that 2%, there are no error bars on this, but 2% is probably below our reliable measurement threshold. The key difference between these environments is these environments here were all subject to a severe drought plus heat stress, whereas these were all higher yielding, more moderate environments. In fact, this one down here that was showing very little variation was actually the average yield at those sites was over 4 tonnes, which for us is pretty good. So there's clearly a correlation between the severity of the stress and the performance of this particular QTL. But the other thing we can do is to try and tease apart different components. So once we've got the genetic information, we can then try and understand a bit more about the structure of the locus. One of the things that emerged here was that we actually have two conflicting, as it were, QTLs that are operating. One that's derived from what we regard as sort of the more drought-susceptible parent, kukri, which is really related to biomass generation. And in the favourable environment, this was actually a favourable trait, whereas in the droughted environment, this was not. And so you can see here under the drought stress, the RAC875 allele was really dominating. So we have here a bit of a conflict that's developing. In the moderate environment, there were benefits associated with the kukri allele, and in other environments, the benefits associated with the RAC875. But the other important thing that came out was that these are actually separated. So we could genetically separate them in the more detailed analysis. I should have explained that the populations we generated were really based initially on about 800 double haploids, and with a backup set of 15,000 real lines that we were able to use to do the high resolution analysis that really underpins these sorts of studies. Okay. Another interesting component of the analysis is if we start looking at the relationship between environments and performances of particular alleles or loci. So this is a region on chromosome 3b, which we know is delivering a yield benefit, and that yield benefit can be in excess of 15% depending upon the environment. But we can use this information to also classify and catalogue environments. So here are a whole series of environments. The environments are now grouped together based upon the way in which they elicit genetic responses of particular loci across the population. And so we can then ask questions about what the characteristics of the environments were. So here is essentially now plotting a series of markers, a series of alleles across the different environments and looking at what the effect is on the yield. And you can see all these ones up here, we have the allele from one of the parents is positive. In these environments down here, the allele from the other parent is positive. So this is up to a 10% yield benefit. If we have, in this case, a kukri allele, this is about a 15% yield benefit if we have the RAC875 allele. So again, we have this conflict between the alleles in the different environments. So what's different between these? This is a straight drought environment. These are all drought plus heat environments, and these were essentially higher yielding, should we say, more comfortable environments. So we actually have here a locus that changes its effect depending upon whether the plants are exposed to drought alone or whether that's in combination with heat. Clearly, this information becomes quite critical for the breeders because it has a big impact upon how they go about deploying this variation. It's not, under all circumstances, going to be beneficial. Okay, so how do we then start combining this with omics data? So to try and tease apart what is happening at the molecular level or at the pathway level within this material. As I said, we basically threw pretty well all of the omics platforms. It's quite hard to do this because, again, if we're looking at drought stress, what kind of drought stress are we looking at? So we tried to model essentially a cyclic drought, which is a fairly typical drought that we'd see in a Mediterranean type environment, where the plants are exposed to quite a severe period of drought, and you can see these 3 time points, they're entering from moderate to severe drought. We then re-water, and we can then look at the ability to recover from drought stress, and then this final time point here is looking at direct recovery. So we're actually measuring a number of different things across here, and when we start trying to tease apart the omics platforms and omics information that's generated, we need to do that from within a context of understanding what the plants are going through at those different phases and stages. And really, just at a fairly simple level, we can then do our comparison of the, so this is looking at transcriptomics data from an RNA-seq experiment. We can then look at the relationship between the cultivar, the treatment, by the timing of the interaction in the process. So here in these first stages here, we're looking at transition from mild to severe drought, from no drought to moderate drought, from moderate drought to severe drought. Here we're looking at recovery and entry into a second drought phase, and here we're looking at direct drought recovery. And we can see that clearly different genes are turned on and respond in different ways over these different pathways and processes. So where do we go with all of this information? I thought maybe the easiest way to illustrate that is with an example again. And this is going back to that 3B locus I showed you before, that curious one that was switching effect depending upon whether it was drought alone or drought plus heat. And so what's quite nice about this region is that chromosome 3B in wheat is the only chromosome that's almost, well, actually has now been fully sequenced. Well, fully sequenced in that there are still a few gaps, but it's not too bad, by the group in Clermont-Ferrand and INRA. And so we could basically then look at our region in a lot of detail. We have, from the mapping, we have it resolved down to less than 0.1 of a centimorgan. As you can see, there are still a lot of genes that sit within this region, but we can then start using the database and information that we have. We have extensive RNA-seq data now from all of the parents, and we have also shotgun sequence, whole genome sequence information on the parents. So we can basically ask questions about whether a SNP is present in any of these genes. So all of the ones labelled here in blue, we see a SNP within the coding sequence when we compare lines that have different versions of this. And I should actually say here that these, the RAC875, Drysdale and Excalibur all have the drought plus heat allele. Kukri and Gladius have the drought alone allele, if you remember back from that previous diagram. I hope this is not getting too confusing. You still with me? Yep. Good, because I'm starting to get confused myself. The ones labelled in red are actually missing between these parents. So we have some genes that have a SNP in the coding sequence. We have some genes that are completely missing. And then we look at our transcript data. We have one gene here where we see a major difference in expression between these groups of parents, depending upon whether they're exposed to drought or not. So, you know, as a result of all that, we end up with quite a long list of possible candidates. And so we're then faced with the next problem about how we go about then sorting out which is the actual gene that underlies this, or if we indeed have the gene. Because remember, we're reliant on a sequence that was generated by from Chinese Spring, and we have within wheat a huge amount of presence or absence of genes across the whole genome. So this really brings me to the next problem that we have, which is again another phenotyping problem really, and that is how do we go about deciding which genes might be controlling our stress or process. And you know, obviously the easiest way to do that, and in many respects the most reliable way, is through transformation, through complementation. To do transformation and phenotyping though for these kinds of traits is also quite difficult. I showed you before that trying to look at correlation between the field and single pot experiments for drought tolerance is essentially a waste of time. So for the GM, we've gone to this what we call a large bin system. So the area of each of these bins is about 1 square metre. They're about 70 centimetres deep. We have quite a detailed complex monitoring system and irrigation system across here. And this was really designed by Boris Parent, who's now moved back to Montpellier, and this is Boris here. And so we can use this system, and you'll see it's also in a glasshouse, in a contained facility, to do the preliminary evaluation of GM lines to try and work out whether we're seeing a drought response or not. We're still at the early stages, so I'm sorry I can't basically say this is the gene that's controlling that, but we have now a whole series of those genes in transgenic lines, and they're moving into the evaluation phase. For other traits, the process, went on. Okay, I seem to have lost a slide here, that's all right. For other traits, we can do more effective screening using other platforms. I won't go into that now, but we have now a number of field sites that have been set up where we can do field trials now for GM lines and hopefully tease apart some of these traits more effectively. All I've talked about so far has been looking at the phenotype and the characterization based upon the, really mainly based upon yield and looking at yield components for our analysis. We know, of course, that certainly when we start talking about drought stress and response to drought, the behaviour of the roots becomes really quite important. And one of the biggest problems we've been trying to tackle in teasing apart what's happening with the roots is how we go about doing the measurements effectively. So I thought I'd just mention a couple of new systems that we're trying to use to tackle this problem. And the first of these is really based upon using DNA, again a genomics platform, to try and work out where the roots are in field trials. So the system is basically, within a field trial, we take soil cores. From the soil cores, we extract the total DNA, and then we quantify the amount of root DNA that's present at different depths down within those cores. This is a technique that was developed by Chunyuan Huang, one of the nice things about it is you can do real measurements within field trials. And it's actually taught us quite a lot about what happens to these varieties in these field trials and under these field sites. You can see here that in a droughted trial it can become quite difficult to take the cores. We had the student who was doing this work originally was a semi-professional boxer, and so she was actually able to do this, but she ended up destroying about 2 or 3 cores a week during the process. So it can be pretty tough, not just getting them in, but getting them back out again. This is an example of the sort of results that one gets from that. So this is essentially screening at 5 different field sites, a series of varieties, and really looking at the surface roots. So looking at the distribution and the viability of roots within the surface 10 or 20 centimetres. And firstly, you can see that in the low-yielding sites, we have very poor root development generally, but we see a huge genotypic variation in the way in which the plant's responding to drought stress. And we can see some lines, for example, up here, that are able more effectively to maintain their roots under different levels of stress. I should say that the position of these points over here is correlated almost entirely with yield, the lowest yielding to the highest yielding site. So we can really now, I think, start teasing apart what's happening to the roots within the field. We can identify germplasm that are better able to maintain their surface roots, in particular under drought stress, because we know that's a key trait in our environment. And we can, and have now started doing the genetic mapping and genetic analysis of this. In addition to that, though, we also see significant changes in the architecture, the internal architecture and structure of the roots in response to drought stress. These are those 2 varieties that I talked to you about before. Remember I told you we threw everything we could at these varieties to try and understand their physiology and biochemistry. And you can see a very different structure of the roots, particularly significant differences in the way in which they respond to drought stress and the way in which they essentially modify their roots in order to deal with the drought stress they're exposed to. Again, these are traits that seem to be quite highly heritable, and so we've also been able to integrate them into the mapping analysis. Given the importance of roots in these changes, that also then raises questions about how we go about applying some of the omics platforms to analyse and characterise this material. And I just thought I'd just show you this slide from colleagues at the University of Melbourne, particularly Uta Rosner's group, who's been looking at using essentially spatial metabolomics. So under this system, they can actually take a root, and this is a root of plants that were, of barley plants in this case, that were exposed to salt stress, and they can then essentially look at metabolites within particular regions of the root, and then group those metabolites. And so we can see there are some that appear to accumulate under salt stress at the tip of the roots, others further back, and some still further back here. And this group here are really right at the very tip of the root. So we can also start asking questions about how we have metabolite shifts in particular regions of the plant. And this is a form of assay that we think may help us make better use of some of the metabolite information, which is really hard to interpret otherwise. Okay, so to return really back to the point I made at the beginning about, can genomics help? I'm working on the assumption that most of the people who would come to a genomics conference believe that genomics is useful and will help. So to some extent, I'm probably preaching to the converted here. I hope I've shown you that there are really quite a lot of ways in which we can utilize some of these platforms and technologies. But there are some really significant issues and problems that we still really need to deal with. And probably the most important of those is really knowing what it is we're trying to measure and how we're going to try and deploy the genomics information to understand those traits more effectively. But certainly, I hope I've shown you that we can actually build resources that can be very useful to support gene isolation and characterization and to give us knowledge about pathways and processes. But I think it's also important within this context that we're working, certainly in the case of wheat and barley, we're working on modern varieties that have been intensively bred over a long period of time. Any new technology we develop and deploy, we have to demonstrate that we can offer something over and above what can be achieved by conventional breeding. There are some major issues still related to the way in which we deploy alleles. And again, I've shown you, hopefully, examples that alleles don't necessarily always go in one direction, but you can have flips that can be quite significant about how you deploy that information. So thank you for your attention.
Next questions. Peter, you said that your platform was not good for drought between the field and the platform. But is it that the platform was not good, or that what you wanted to measure with the platform was not sort of well designed enough? So in other words, the question you were asking to your platform, what trait should be measured, was it sort of well conceived? Or maybe the platform itself was not conceived in a way that it would address your questions?
I think that certainly we can't eliminate that there may not be other measures that we could do within that platform. There may be other components of the images that are being collected that we couldn't analyze that would give us a correlation. Personally, however, I think the fundamental problem we have with drought stress is that we're looking at essentially in wheat a set of genetically identical plants that are competing with one another for moisture. And it's really that competition between plants that's quite crucial. What we're trying to do now within the platform is to see whether we can grow multiple plants within those pots. We're quite constrained in how large pots we can grow. From previous studies that we've done, we know that if you try and correlate just grain that you harvest from plants grown in pots, there's virtually no correlation until you go to more than 3 or 4 plants per pot, so you have some level of competition. That's why we're going to a large bin system for the GM evaluation. Maybe we can get better correlation if we start using 3 or 4 plants per pot, but then the image analysis becomes more complicated and difficult. So you're right, I can't say categorically the platform is no good for that. I can say that the way we're using the platform at the moment is not suitable.
Okay, due to the time reason, we have to hold our further questions at the end of the session. That's again, thanks, Peter.